old version first. Okay, if you have a difficulty to find the place where you found the toolbox, you can uninstall the old version. If you don't know, let me know. Then from here, you should have this clean and is ready to upgrade to version 26.2. Okay, then you can run this and check whether you have the latest version. Okay, so then check. Okay, this is version number 26.2. That's the updated from last week. We need that because in this network analysis, we have this cell-cell communication package, SC10 XCT. In older version, I only have Python-based, but for this new version, we have MATLAB-based. It's not fully implemented, but there are two options make your analysis much faster. So if you don't need very accurate results, maybe just run this very quickly. I'll show you later by the end of this second half of lecture. Okay, so we have this upgraded. Then come back to our Teams channel. So this is what we are going to do today. We will look at this SVM function in MATLAB using examples to run a two-class SVM classification problem, and then look at this decision boundary and where the support vector points are and the margin. Just follow the documentation example. Then we will take a look at this fully functioned support vector machine options. So this is one of the most important simplified version of this, but you can expand its function to adding kernels, for example. Then we are going to play with a small illustrative example called a visualize projection to understand how this kernel function works. Then we will move to install Python in your computer in order to have this MATLAB Python cross-language communications, and some of this function like single cell 24 XCT, the neural network part is implemented in Python, so I still have to rely on Python. So then we have annotation for cell type, and I believe 2 weeks ago we showed this function very quickly. But now we revisit this capacity in the toolbox and see if you want to identify the cell type. On marker gene database, then how can you annotate your cells? Then we will play with this example to look at ligand receptor communications, visualization. At the end, we will read this paper together. So this paper is called Articles to Code. The idea is you can give ChatGPT or other language model this prompt, like, please read the paper in the PDF and then implement this method in MATLAB. So language model will read the paper and write the function for you. So that will be your homework, and then you need to find the right paper. For example, this is the paper I found as an example. If we have this paper, we download its PDF, upload the PDF to GPT or Gemini or Cloud Code. Let the language model to implement the result, the function. Then you need to know how to test in MATLAB environment whether this function is fully implemented correctly by the language model or whether I need some changes. So that will be your homework. So that's the last part. Okay. Any questions for this agenda for today? We're okay. So let's click this link and to visit documentation page. So this is a function for support vector classification algorithm. You can get the same thing from here, and if you feed, so you know this function, which function is located in the statistics toolbox with a class regression, and the function is called feed C SVM. Then if you do documentation doc feed C, that's the same page I just visited. So for classification applications, you need input data X, big X, or you want to put into a table. So MATLAB have a table structure rather than just a matrix. Table is just a matrix with a annotation for the column and the rows. Then you need a Y, which is each sample's label classification, because you want to train your SVM machine to get the model, MDL. I hope you have the picture, because classification is supervised algorithm, so you need input data to do the training. So this part is doing the training. You have X, you know each sample's label in Y. Y is a vector. Okay? So you train the model to change the parameters, to find the boundary, find the support vector, and then the model will be able to use to predict any other given data from your input later. Okay. So this will train the support vector machine model for this binary classification. Okay. Let's move down, and there are different ways to get data into it. We will just use very simply simplified coding, like here. Okay, you need a input X matrix and class label in the vector Y. Here is an example. You have seen this before. This is the fish example. So what do you need to do on your computer is just click this copy button here. Well, let's see. That will get your whole file. Maybe let's select and paste. Uh-huh. Oh, yes. Copy command will give you the whole. So not so clear. Maybe we just select and copy. You have full control for everything. So from here, I just copied the code, so we get X and Y. It's a fish species with two variables to describe each fish. We have 100 fish and their labels. Okay, so that's what we need, X and Y. Come back to this page. And you copy this. That's all you need to train this model. Okay, this is a very simple model. X and Y, okay? So the output, what we call a model, is a trained classification device. It's a classifier. So it's SVM-based classification classifier. And the response name is Y, that's your Y. And in the Y, they have two different species, fish, and those are the parameters of the training. Okay. So what you can use for this? So if it already trained, if you do a dot here, you can see the function like a predict. Okay, because it's already been trained, and you can use the function inside this model provided. For example, you gave the first fish it should give you the answer. The first fish should be this. Okay. So if you give a new fish, which two numbers, 0.5, 0.3. So each fish has two values. And this fish should be classified as a versicolor. If you have, say, 2.5, this may have the same fish. So we don't know how this model has been trained, but the two fish I test. I set up two fish and by random numbers, and they still be classified versicolor. Okay. So that's the trained model. Let's look at, so you can look at the properties of this trained model, and all the information has been encapsulated inside this model structure. For example, here you are not asked for the prediction function, you ask for what's the class name. You have two groups of the species. This is the variable name. And here is the extract those important points. Okay, you extract support vector. So let's see what's in the SV. If you extract this support vector out, this is supposed to be some data points. Okay, SV, there are 24 points. So those points are the boundary of this separation. Okay, have been used for the training the model. They from the input data. Input data is X, so we have 100 X input data. Some of those are more important than others. So those support vectors are the important samples. We can copy this part of the figure to visualize. So what we will see here is first, we plot all the data points, all the fish. So you have X, that's 100. And then we will plot support vector position with this black circle, which is black circle are the 24 support vector identified by the algorithm. So if you paste this piece of visualization code, you get this scatter plot. So scatter plot, you have X axis, Y axis. Those are the two numbers you had from the sample itself. Then, because this the grouped scatter, you have color by the Y. Okay, so this is a grouped scatter. You have Y to indicate two different groups. Worst color are those blue ones, and Virginica are the red ones. So if I had input with a sample, say 6 or 2.2, then it should be the Veronica. So if I do model the prediction, see, I did remember I tried a couple of different fish, and they all being classified as a versicolor because I don't know how to set up. And if I set up as a 6, 2.2, I know this should be giving me a Veronica, because I look at the scatter plot. I know if I have a new data points, it's 6 or 2.2, it should be grouped as a Virginica fish. So this is a visualization for your training data and which training data point has been used as a support vector, so from the machine algorithm. Let's move down and then to draw this decision boundary or margin. Yeah, I think we need this part to get the beta and the B. So these are the slopes and intercept for drawing this separation line. we had that. Let's plot the lines. Hope. Yeah, so for this in this case, maybe we should restart by copy this. So for this part, we are not going to copy each separated code segmentation. Again, we just directly copy everything. Okay, copy this, and then come back to here. We have all predefined this. Annotated documentation. We can run each section. You highlight this section, then click this. Okay, this has been loaded. And then the second section that you run will be trained. This model should have so this is like fresh. Yeah, if you delete this, if you clean this output and run that again, the output will be input inside this documentation rather than pop up. So that's what here they use a slightly different input example, because the one we just had is not very clear. They are not separate, not giving you this margin. But this example, they also two different fish. This time they use Virginica and Setosa. And the reason I believe they use this is the scatter plot. They're more separatable to let you to see this boundary. So that's what the boundaries have been found. So this is the separation line, and those are the support vectors. And then you have this margin, upper and the lower margin have been found. So you can take a look at these parameters, how they extract from the training model to get this margin boundary to draw these lines. You can see this will be a blue line with a dash. This is a plot, okay? Blue line with a dash. This is red line with a dash. This lower boundary. Okay, so if you want to spend the time, you can run this again offline sometime. So we are going to move on to this page, to read more documentation on the support vector machine. This will look like pretty much like lecture we had before. You have a separatable data, and then you decide this separating line or hyperplane, depends on your dimensionality, and then to find those support vectors, and then try to maximize the margin between two different classes. So that's example of the separatable data. Then you try to find this beta parameter or coefficient. This is a slope of this line, and then you have this intercept. You need to compute the distance between all the data points to this line once the beta and the b are given. So you need a way to compute this. Remember, at the end there are dot products. Then you found the support vectors and start to optimize the slope and this b and beta and b to get this maximized margin. Okay, they're non-separable, so SVM will use soft margin to tolerate some of these errors, which terms here. Okay, you want to see visualization, but that's implemented inside this function. Soft margins. All this setup rely on optimization. So this is like a search engine to find the combinations of parameters. So which value I should give to in the beta coefficient, what value I should give to b. So they all rely on this solver or optimization of solution founders. Okay, for the nonlinear transformation with kernels, the idea was like this. Okay, so this is G is a kernel function. So you apply kernel function to your data points. Any given two data points pairwise. Then this result is equal to you transform your data, then get a dot product between these two original data transformed. So this relationship gave you a shortcut using kernel function directly applied to raw data before transformation, and you get the transformed data dot product. And we know dot product is very, very useful anywhere in the machine learning. Okay, so if we can compute this very quickly, we get operations in the transformed high dimensional domain without even have to transform. So we don't even know what this function, this mapping function is. We only use kernel function to do this computation for the transformed data's dot product. So those are the commonly used kernel function. So most of this people use Gaussian kernel, so G. So you can have a capital K as a kernel function, and you compute two given points, and basically there are distance between them, and then take this exponential function. Okay. So this is a kernel function, which is Gaussian kernel. So for each kernel function, you're supposed to have a mapping function. Okay, so you need to prove your kernel function results the same as your mapping function applied dot product of after your original data is mapped to high dimensional. So I rarely see people discuss what is a mapping function for Gaussian kernel. So that's why I visited this GPT and light language model to see, okay, for the Gaussian kernel, so that's the formula, right? Then we normally don't discuss the mapping function, but this is a formula for the mapping function. Okay, it's implied there. There's a mapping function in there. Okay, so let's go back to the documentation. So in the SVM, the easiest way is we use this X and Y as an input. We just did that. There are optional variables. So if you decide, say, okay, I need to use kernel function to transform my original data into X, you can select this optional variable to give to instruction to this support vector machine implementation. You see, I want to use Gaussian kernel. So then you just select this function. There are other functions. So whether you want to standardize your input, if you put a true, it will do that. Okay, so this, if you have non-separable, non-linear, you can apply this non-linear transformation by invoke this kernel function, allow it to do this kernelized SVM. So not as an example we had here. This is definitely separable linearly, so we don't need to do the kernel transformation. But if your data is too complicated to make clear linear separable, then you need to invoke to call this kernelized SVM function. So that's the way it's implemented. So once you have this trained model, as I said, you can use the model dot predict, or the predict, put the model here, then your new data in there, the same thing, and you get the label prediction. There's some other steps that you can refine your results. Okay, there are many options. Kernel function is one of the parameters you can give to the SVM. There are others. Okay. The result can be given to another function called cross-validation. You do the CV to test, to give you an idea whether your trained model is robust and what's the accuracy of the test. So this is an example for using Gaussian kernel to do the training. So let's copy. Yeah, let's copy this to set up some example data. So now we can close this. And I will clear my workspace. copy the code I just get. So this gave you a data, 100 data points. Then copy this, gave you data 2. So we have two sets. We have two sets of data generated. Then you copy this to plot these two sets of data you just simulated out. So you get this because we simulate the red dots first, then blue dots. So this set up two classes, definitely not linearly separable. So for this data, we have X, the input will be data 1, data 2, and then we are going to put together, so put them together, data 1, data 2 as an input for the SVM training. Then you have a label because you have 100 data type 1, 101, 2, and you set a class that first 100 as a -1, the rest as a +1. So this place is what you set as the input for the training. So we copy the code over there and then paste. Now you have your data 3. Data 3 has 200 samples, and their label is in the class. The first 100 is -1 label, and the rest are +1. So that's where we set up the data. Now we need to give this X and Y. X is input data, data 3. Y is the label for each sample. Now we need to call kernel separation function. Okay. So now we train the data that gave the result as a CL, as a classifier. Okay. So that's we can copy all the code here very quickly to the whole section. Come back to here. Yeah.
Exactly did the step do?
Which step?
What we just pasted, this code block.
This whole block.
Yeah.
The whole block will use the kernelized SVM, the function, to train the models, and you get a CL as a model. The rest of them just plot the,
To get the decision boundary.
yeah, to get the decision boundary, yeah. So you really don't need to look at the rest. This is just for visualization purpose. So you trained your model, so you can use CL to classify your data points as a +1 or -1. It's a kernel version. But this gives you the visualization for what are the kernel points with the black circle. Black circle, hardly see, but there's this one, they're black circles around. Yeah, you can see this support vectors. This, that. Okay. So this result is slightly different, but it's still kernel version without the fine-tuning of the rest of the parameters. So the classifier number 2 trend gave you a slightly different solution. Maybe there are several misclassification. You can define your own kernel function. So here is an example. Then the example is the job is to classify these points. In this case, the Gaussian kernel will not give you the very good solution. So if you define your own sigmoid kernel function, like something like this, okay, you provide this as a function to give the kernel variable options, then the model starts to train based on this kernel function and get this boundary. You can refine your kernel function to improve your solution. So if you have no idea which kernel to use, you can try different kernel functions and see which one give you the best trained model. Okay. Okay, I think that's all for this part. All right, I hope you have some good understanding of the whole thing. Now we can clear the space. Come back to this page. So we are moving to item number 3. So this we will open a file which is a source code for MATLAB. And then, so we do this before break, okay? So this is a place you can click to get the source code for this. You don't need to do anything, you just click this and you will see the source code of visualize projection. So you just use Ctrl A to copy the whole thing, Ctrl C. Now come back to this clean workspace. And the Ctrl V to paste the code in the command line. So that's what you will get. Don't move yet. So now your cursor, your mouse will become this, and now you are going to use your mouse to click to create some data points. Mac machine. Mac has a problem. Okay. Then you just look at what I'm doing here. Okay. Okay. So this is just a small demonstration of the kernel transformation. So this allows you to, by click, to create sample points. Okay. So I just click and create this random sample. And then once I finish my red sample, I use the mouse right click, and it's done with the red circle. Then I start to put the blue points in the center. So So once I'm done, I just right click to give the last blue point. Then mouse right click and the program just finished. The program will just find the center of your sample and then do the kernel transformation and show you the 3D separation. Now you can separate this. Okay, so that's the whole purpose of this. I never tried this on the Mac machines. I don't know. It's not working. It's working now. But why did you say not working?
No, no. I mean, there were two. So there were two. One was there in that message that file was the file name was there. I didn't see it.
I don't understand. But it's good. It's working now. So if you look at inside this code, you should see where the hold on, and G input into to get your click. And once you finish this, this is where the kernel transformation. If you look at this function carefully enough, you will recognize this is the kernel function is Gaussian, which is the formula from same as what we described here. Yeah, you will recognize this formula here, okay? Exponential minus negative. That's where this is. Okay, let's take a break. Okay, so we will continue. In the channel here, we also have this PDF. It's a user guide for support vector machine. And this is very technique. Documentation here. It's like a more formally described. This linear, nonlinear classifiers. I hope at this stage you are not scared of those commonly used mathematical formulas. For example, this is a kernel function, take two different data points in, and this is the mapping function applied to each of these data points. And you know this is the transpose of the first data points and this. So together this is a dot product. And then you start to recognize those. So this will be a function, and you have your alpha and the b, and this is a straight line. So I hope slowly you start to feel comfortable with those expressions. So there's a norm of this w vector, so how long it is, basically, the 1 over it. This is the L2 norm. Yeah, I mean, sooner or later, machine will replace you. You can just give to language model to let it to understand, but it's better. You know what's going on. So just hope you are not scared of from this expression of those formulas. Okay, let's move to another activities. We finish the visualization. Now, let's try to set up Python environment in our machine. So the first thing I will do in this machine is, so every machine will be slightly different. Like, let's try whether I have, from my command line, whether I have Python installed. It's pointing me to Microsoft Store. So this is not the version that I have a fully control. So I don't know where this will be installed, something like that. It also may require admin. Maybe I should give it a try. Oh, the only problem is I don't have a control for the version of this. So this will install. Which version? So I really need some control for the which version I installed. So this may be not the best solution. Let me look at the software center, see whether we have Python in here. Okay, I have several options. We have a 3.12, 3.11, 3.13. I don't know on your machine, so you have your own control of your Mac, your laptop. That's good. So use this if you have Windows machine to install Python from a Microsoft Store. This is very convenient but I don't know where you can control which version you install. In the university machine here, we have a software center which you can choose which version you install. The third option will be follow the instruction on my here is to install so-called mini codon. This environment allow you to select a different version. So for all of these three solutions, we need to look at this requirement, which in the link A items here. Let's just click. So this tells you your MATLAB version will compatible with which Python version. We are using the latest MATLAB version with 25B, so it's comparable with Python 3.12. So we cannot install 3.13 because it's not comparable with our MATLAB. Okay, so once I get this information, I know I can install 3.12 for my Python version. So I will come back to my software center here by click this version. So it's already there. So I've already installed this, so I should have but because I don't found this Python I installed, so I will just go ahead to install from the Microsoft Store here. Click Get. Yeah, so have some problem with this because this only allow me to install the 3.14 version. It's too new for us. It also requires the admin power, so I can't do this. So I gave up install from Microsoft Store because, oh, now it's coming out. So let's see where Python installed. Yeah, I don't know what's going on here. So some installation has been here because I want to install this version from a software center. It's already installed, but I wasn't be able to find where it is installed. But now come up with a solution how it is set up. And I had 3.12 Python installed, which is located in this place. So that's the information you need to set up. You need to on your machine. So if you have a Windows, search for where your Python is installed and which version you had. Yeah, on Mac you have it. Yeah.
When you use the install through the Windows Store, the SGA2 doesn't pick it automatically. You have to go into this folder, and when you try, you get an error. But when you install Python directly from the Python website,
Yeah.
then the SGA2 picks it up by itself.
Okay.
When you go to the Windows Store.
Okay. So maybe this is something special, not as a clean installation. You tried yourself. Okay. So I think Felix provide another solution is to directly install from the Python download. Yeah. So if you see in Windows install Python, it will lead you to this official website. That's where you install. Yes. Okay. So this, I think that's the best solution. So from here you just go to that version, then install. Yeah, 12.3 also works. Okay. So assume we just do this and without bothering through the mini codon. This should be okay. So we don't need this. Okay. Because we can install the Python itself directly. Okay, assume I have that installed. I will try, see whether I can set it up easily from this toolbox here. So I just need to know where I installed and point to my MATLAB to that point to know where to call Python. So it's from here, and you get external, then set up Python environment. Python 12 already installed. If I use, yes. So you just need to find where you installed your Python, then you get. So that's where I already have set up. Where is it here? Yeah, so if you installed in a program, you should have that in there. Okay, 3.12. I just select that folder and point to this. That's all we need to know. Let me know if you have a problem. I don't know in Mac machine what's going on there. So we can test with some example data. So if you do this, label the data with batch ID. Okay. Then from external, we select Python tools. these tools, including batch integration, so this is called Harmony. So let's see if we had this set up, whether we can run this Harmony application by directly click this. So we need a working folder. So let's see your documents, that's fine. Use this. Program. Okay, so this is the first time I run Python version of the Harmony. So in the background, the package tries to install itself on this machine. So the first time run may take longer. I'm still not so sure whether the package will be correctly installed, but we just wait and see whether this will be finished. Errors. Yeah, so there the problem with this function here. Okay, so I will take a look and see whether we can fix this later. But this is just the way we are going to do the calling Python functions. Let's try a different one. I will run this again and see whether but I yeah, I was thinking this because the first time run is not giving the results, but I just try that again and still have the same issue. So that's a bug which will be fixed later. So let's try a different Python function. Detect duplicates. So also this first time this package will be installed. Not correctly installed. Try again. Yep, some issues. So there are some issues with the installation, and for unknown reason, Normally what you can do is to install from this Python environment and see whether you can solve the problem. For example, if you had the Python version installed in your Python environment, it should be run this. Okay. All right. So we don't have many functions that exclusively in Python, but you need to set up. You need to know what they are doing. I can found a way to fix some of these problems. So as I said, if you know how to re-implement some of this function, for example, in MATLAB, so we are trying to do later on, then you can get rid of these troubles in communicating between packages. You have a problem installation of Python and different version, compatibility. You pass on the parameters, and the results may be not successfully. So there are issues, and I believe there are better solutions using this web coding now. Okay, let's move on to our next annotation for this. Okay. So I'm going to use example from the item number 6. So let's just click this sample ID. So we are going to download this sample to our MATLAB environment. Then we try to annotate this sample using standard approach, as well as you have a customized marker database. Okay. So now come back to here and to click this import data, and then geo accession number. You just copied the one is GSM 3121363. So this is mouse skin data, maybe. Skin wound tissue cells. Okay. So for the customized cell type annotation, this is applicable to the situation where you know what cell type in your sample. So you know your marker genes as well. So you see, okay, I don't rely on public data information. I just want to use my own knowledge to annotate these cells. I know they're fibroblasts. I know they're keratin sites. I know they're some immune cells. So we can put those names and where they're not markers. So you can build your own customized annotation tool. Let's first do a QC for this data. So you just QC filter basic QC. Cannot be redone because stringent. Okay, so this clean data, it contains 22,000 cells, about. Let's do an embedding. Okay, 3D Disney. using highly variable genes. Then we will do this classification, clustering, and then we will do annotation for each small clusters. So clustering is this button here. Okay. Based on this embedding results, then do k-means. So now I'm going to annotate each of this. So if you don't want to use your own annotation marker genes, you can just run this sun cell type 2 groups. By default, we will use PangLawDB, the database information. So now you are going to annotate this cell type by yourself using annotation and customized marker genes instead of PangLawDB. PangLawDB is relatively old. So now if you decide to use your own, then here give you an option. There's another marker gene database. It's called a single cell SC type marker gene. So if you want to use that, you can have a preload, so you don't have to type yourself. If you don't use this, if you click no, then you have to have your own marker genes and the cell type annotation table. Okay, so now for demonstration purpose, I just do this. I will load the cell type, SC type marker gene list. If I say yes, you will find this predefined organ and tissues. I need to find, there's no skin, so it's very limited. Which one should I use? Use muscle? Use brain to annotate your skin? Muscle should be close, yes. So if you select the muscle, this will be preload your markers. Okay. So if you take a look, this very simple. You have a cell type, then you have a tab, then your genes. Okay. So I just go ahead and using this customized to annotate my cells. I can see from the background that there are a lot of unknown cell type because none of your marker genes support those small clusters belong to any given cell type you proposed So this is an indication that your marker gene database may be not a good setup. So you really need to understand your sample better than this. Almost there. So I want to merge this because there's no, make no sense if you want to annotate each separately for this purpose. So that's my annotation. A lot of, so yellow ones are unknown, a lot of cells unknown. There are some lymphatic endothelium cell in the middle. Then you have this mega karyocytes. Okay. Okay, so just to show you how to use this annotation, which use your own marker genes database. So I think we have to resort to using PangalDB to do the annotation. Mouse. So PangalDB gave us something like this. This is based on the annotation of this unknown, and so I think we need to redo this class string to let PangalDB to run on each of these small clusters again. Okay. So we are going to use this annotation to look at ligand receptor genes expression. A set of this selected, the name of the gene are given in this. So we will look at the receptor gene PDGFRA And then this is CAV1 as an example. There are many different pairs you can look at, but I hope this two give you some idea. Okay, this annotation is done. We just merge all the subclusters into the same. Okay, we get some T cell, endocytomes and macrophages. Okay, at this stage, I want to save into the file, so just in case, we can load it back later. So I will put into my document. Okay, so how can we visualize the expression level of the genes? So these are two genes I just copied, gene name. We check from this first button here to select gene, see whether these two genes are there. So I do a search. The gene name I got from the database is from human, so you see the all capital gene name, but for the mouse, you have a first character is capitalized and the rest of them are not, but there is a homolog in mouse, so I can select this gene. Then the second gene is called CAV1, and I also have this, so I just click. So these are two genes I select in order to run, to look at their gene expression. I want to look at the raw UMI. So we don't do any normalization transformation, it's just untouched X. So in this case, I select no, just want to look at expression level. Then you will get, for each gene, you get two panels. On your left is this heat map in 3D, and then those indicate expression level. And here is a 2D with a vertical bar indicate this gene's expression. The first thing normally I will see is how many genes, how many cells express this gene. So this is 18.9%. So there are 4,160 cells in our sample express this gene. And they concentrate in fibroblast here. And then look at the second gene. Look at this gene, seems highly expressed in this particular subcluster. Okay, it's right here. So we'd better to know, okay, if there's communications between this. It could be between these subclusters, this subcluster here, to this center, right? Okay, for this we need to move back. So I need to find a way to identify which cluster I'm talking about. So it looks like it's this one here, this group. It's donor cells. Yeah, so this group here. So I need to go back to my annotation and see which group. Sometimes it's difficult. So which group is this? So this group looks like endothelium cell. So I'm highlighting which is this group. So this communication is between endothelium cell to fibroblast, which is the center part of this fibroblast. Okay, so this is visual evidence, right? I hope you are following me what is going on. So this is from endothelium cell. Not which one it is exactly. It's from this part. You can see it's a very high bar. And endothelium cell provide this ligand and gave to this group of fibroblasts. Okay, so that's a signal I can see from the pairwise this gene expression and the corresponding cell type. Okay, so now if you have no idea, see, I clean my workspace. If you have no idea which pair to look at, there are several options to allow you to do this cell-cell communication analysis. We can use so-called one sample single cell communication. Okay, that's what we are doing. Let's you can also use this method, which is unpublished. Let's try this method first. This crosstalk. Because this is a lightweight method, so you can apply this to all the cells, all the genes. That's a lot of genes. If you do network-based, it's really time-consuming. So I'm going to try this first. Then you need to select which cell type you want to play with. You need two of them, so that's between these two. So this method will help you to identify those pairs, ligand receptor, okay, the top ones. So this very quickly give you some idea, okay. This could be the pairs which you want to focus on. So I gave a table, look like this. then you can save or to Excel tables. So this is a method. If you decide to use the network-based, so what I'm going to do here is to try to remove some genes. So I will select the first items here to remove those genes that not very informative at the end, because they are never been discovered as a ligand or receptors. So I can clean this, for example, the mitochondrial genes, hemoglobin genes, ribosome genes. So I know they are not very useful. So if I click this, and the program will select these genes with a name or expression, very informative. To accept this, now my genes only have 3,600 genes left after this treatment. So now the result will be more informative. In that case, we can start to do, I will do a while filtering first to see whether we need to remove anything. Okay, so clean data. Now you can use the network here to one sample, 24-net XCT, then continue. You can select, for example, the spectral plus spearmint. So this is a very quick method. Our Python have not installed yet, so I cannot select, but this one, you can select this and then select cell type. Again, it's between fibroblast and the endothelium cell. Then select the direction as well. Then the program will build a network and then save the results into a folder. And this is a place where you get the result. Okay, so that's a ligand receptor pairs identified using this network approach from endothelium cell to fibroblast. Okay, so that's the way we can do. So the last, you need to go back to read this paper. So this is a bioinformatics paper. Try to use large language model to implement the algorithm from reading the paper, like language model to reading the scientific paper itself directly. So the authors use several examples. They tried to implement the random forest and the combat. Combat is bunch effects remove algorithm. And another program called Augusta, and also the GC. So they tried a couple of different papers. Okay. So they basically just upload the PDF of the paper, but implement that method, and then give to different language model to see which one is best. So you don't have to test different models. You can just select the one that works best for you. If you have access to GPT 5.5 5.2 or other models, use one of these. Okay? So you have different options. Gemini 2.5 Pro maybe is a good option as well. All you need to do is just find a paper which good for language model to implement. So this paper has to be very clearly described how this method will be implemented, what the input, what output. If you have a review paper, then it won't work because there's no methodology, no expression, no pseudocode, no formula. So you can take a look at how they did. And they found the paper described the combat from this 2007. One example I gave that you can use is from this paper. If you click a link here, is a paper which to use Fourier transform to generate, synthesize new single-cell data to do the simulation. So if you download this paper, you can see the methods are very clear and how the information will be transformed, what's going on. So this will be comprehensible for language model. So you upload this PDF to language model, and then you need a instruction, the prompt to see, okay, use only the information in the attached PDF, try your best to create this algorithm in MATLAB, that exact match. Okay, so if you read the bioinformatics paper, that's what the prompt they designed for. They just use this instruction. This is the instruction to let the language model to implement combat, to implement random forest, get some success. So your homework will be find a paper which something looks like this, have a very clear description of the methods, step 1, 2, 3. And you don't have to understand this, you just give to the language model. But you need to know what's the input, what's output, so you can test, you can validate the results generated. Okay, so if you are not so sure which paper to implement, send the link to me and I will take a look and see, okay, yes, go ahead, and you can move forward. Okay, any questions? You need to figure out once it's implemented how you test the source code, the implementation yourself. Is that next week will be a spring break? Okay, yeah. So you have a long time to think and no deadline, just shoot me the email. Let me know which one you want to implement. Give it a try. So I will write those instructions in the assignment, the homework assignment. Okay, so this homework wasn't possible last year, so it's only this year. Impossible last year. The language model is not smart enough at that time. Now it's time. Enjoy your spring break and see you the week after next week.